Here is La Belle France, a country of 1,000 faces whose contours and colors create magnificent landscapes and spectacular views. France is the first agri-food sector in Europe. Horticulture is internationally renowned. Discover the French art de vivre with its rich cultural heritage, taste for elegance, fine wine and gastronomy, and a know-how for hosting major events. Host of the 31st International Horticulture Congress in 2022 and recognized as the greenest city in France, Angers has become a leading example in the plant world. The Angers region, located in the heart of the beautiful Loire Valley, benefits from a great variety of mineral resources. Its mild climate and proximity to the ocean create an ideal setting for horticultural production. Exotic plants have been successfully imported to this region ever since the 15th century by King René. In the 19th century, leading names in the plant sector set up their companies in Angers and made the city the greatest plant breeding capital in Europe. Roger de Villemorin, original ISHS president and a great seed producer, organized the first International Horticulture Congress in 1958. Since then, Angers has been delivering millions of horticultural varieties throughout France, Europe, and the world. Dynamic and varied companies have turned the region into the top location for horticulture and seed production in France. This is why the European Union chose Angers to set up the Community Plant Variety Office. In the 20th century, the region became a competitiveness cluster with the creation of the Vegipolice International Network. Vegipolice is here to boost innovation and encourage competition between companies, research, and training centers. Vegipolice is now a worldwide reference for plant creation and production, with more favorable impacts on biodiversity, health, and environment. As the first French campus dedicated to vegetation, Angers is a unique place which attracts professionals, researchers, and students from all over the world. Ideally located in western France, an hour and a half from Paris and close to many European capitals, Angers enjoys the unique European leisure park dedicated to plants and offers top-of-the-line infrastructure facilities. We look forward to seeing you in Angers for the 31st International Horticulture Congress in August 2022.
Hello to all our attendees for joining us for the day 4 of our webinar series about peach production. And at first, I would like to introduce you Francois Laurens, who is the organizer of the International Horticultural Congress that will be held next August in Angers in France. I would like, before starting our scientific program, to give the floor to Francois to provide us helpful information regarding the organization of this very big event. Francois, the floor is yours.
Thank you. Thank you very much, George. Hello, everybody. Good morning, good afternoon, good evening. So thank you very much, George, to invite me to give a few words on the IHC. So I will not speak about, I'm not a pitch guy, I'm working on Apple. But don't worry, I will not speak about science, just a small advertisement about this big event which we held next year in August. So, and of course, I will follow the rest of the day because I see very interesting topics. And the next one is also very interesting, the one from Laura, and I know a little bit what she's speaking about. Anyway, so a few words about the IHC. So for those who don't know, Angers, it's on a tiny city on the western part of Europe and western part of France. So this is an ISHS event, so I don't need to present ISHS. Or for those who, I think you already have the introduction from Peter on the first day of the webinar, so I don't want to enter there. So that one of the activity of, or the main activity actually of ISHS is to organize a symposia. and to organize also Congress. So you see the list of the previous Congress and the one in Angers and the next one in Kyoto. And so just we are dealing with horticulture. Horticulture is a wide term, so from upstream to downstream, including fruit and vegetables, flowers, wine, medicinal plants, and so on. So now about the IHE, so we defined 3 major axes for this event. So the first one, for me, the most important is the high-level scientific event. So we have a strong scientific policy for the Congress. Quite an original point. We want to make the link between the research, the industry, and the education. It's not only on the world. We are organizing specific activities to make these concrete links, and this more on the DNA of ISHS to make to make the link with the north and the southern countries. We are planning, so of course with the COVID things are not so easy. It's much easier for us hopefully than for George for the peach, because we hope that the next year it will be a little better, at least for most of the countries. So because we don't know and because we see that the webinars are very successful, we already plan a hybrid event, but we really put emphasis on the on-site Congress, of course. So we are expecting at least 2,500 attendees. Actually, 1 year ago or 2 years ago, we are planning more than 3,000. So we are a little bit more cautious now from a lot of countries. And the target audience is mainly on the researcher, but we also plan quite a big part of professionals, of growers, and so on. So 20%. On the provenance, we hope that most of the people will come from Europe. We are expecting quite a lot of people from Asia, from Americas, Oceania, and Africa. So just a rough estimation so far. And the program, so it will be held in the middle of August, as all the IHE from the beginning. So it's 1 week. With the opening ceremony on the Sunday, some tours before, some touristic tour after, and the week actually will be a structure per day, starting by plenary sessions. We are working very hard now to find a high level grand keynote speaker, and I can tell you that there will be excellent keynote for the plenary. Then the normal symposia with including e-posters. At the end of the day, you have a workshop, business, or small roundtable, and so this is the normal planning for each day, except for the Wednesday, where we plan local technical tours around Angers. And don't forget the social events. So we really want that it's an excellent scientific event, but also we really want a social thing to happen. So every day, every night, there will be an event in the city. And of course, don't worry, don't miss the opening ceremony. So these are just so in addition to the symposium and the plenary, we also will have a exhibition center with a booth and things like that. We also try to organize B2B job dating for the students, but we really want to have increase the networking between the people. It will be, so we are in Angers. Angers is not a huge city. For those who knows, so the city center is here, and we are in the middle of the city center in the Congress Center, but also using some facilities at the university, which is a 5 or 6 minute walk from the center. Actually, we have 3 sites. And you will be, I don't know, few minutes from your hotels, from all the hotels in the city. And there's a tram which is connecting the three sides plus and also within the city. We defined some main scientific challenges, four actually scientific challenges, which actually the topic of the four plenary sessions. One on competitiveness and skills for horticulture sectors, one on nutrition, health, one on sustainability, agroecology, and so on, and the last one on climate change, mitigation, and so on. So this will be the backbone of the Congress. Plus, of course, we will deal with many very important topics around the horticulture. So these are I will not detail all the symposia. So you can find all this information on our website. They've been updated recently with the exact definition and frame of each symposia, the convener, the scientific committee. So I just highlight or put a circle on the symposia, which of course will be important for some of you on peach. So you have something on the breeding, you have something on genetic resources, on the quality of the plants, on the in vitro techniques. Then so it's not for you, but there are a lot of things, actually, a lot of symposia, actually, 3 or 4 on urban horticulture. Then 2 symposium on abiotic stress, so 1 abiotic stresses and 1 specific on water. Plant nutrition, epidemiology, agroecology. So of course it's important for you too. Innovative perennial crop management, so how to manage the orchards, the fruit quality, and one important one, the S18 about the precision and digital horticulture. So it will be very, very important. Then you have few symposia on specific crops, berry, wine, of course, we're in France, banana, and the colorant plants, plus some on post-harvest, and one on nutrition, which is the 5F, for those who know it, ISHS symposium, and one specific on the medicinal and aromatic plants. So this will be organized all along the week. And the idea is to actually the added value to attend this kind of meeting is to have all these different areas in one place in 1 week. Anyway, for those who knows, so it's not so easy sometimes to manage, but we'll try to make it very clear and very, very easy for everybody. The key dates, so the call of abstract has been open last week, end of last week. We are expecting the submission of abstract in November. The early bird registration start at the end of this month, and then you will follow the different things. You can find that on the website. And we are the ambition to publish the ACTA. So we put 30th of August. Perhaps it will be a little bit later, but very close after the end of the Congress. For example, the strawberry Congress, they succeeded to publish the ACTA the day of the webinar, which is really great. This is the steering committee. So if you are dealing with science, you deal with Emmanuel Geoffriot, is in charge of science. Perhaps some of you know Rémi Cad from CIRAD is in charge of international relationship. We have our honorary president, Jean-Claude, and Valérie, who is part of, actually, we have a big support of the Angers CP, and so they are also part of the organization. And so Valérie is in charge of that. Emmanuel Rousseau is the part of the Végépolis Valley, and she's in charge of the communication. And Marine is managing everything, Michael on the student part, and so on. Okay, so I think I gave you most of the information. Just few touristic things. Of course, you have science, but you have also enjoyed the tourism, so around Angers, but also in different parts of our region or France. So, and we'll try to organize mixed tours, touristic and technical tours after or before or after the Congress. Thank you for your attention. Thank you very much, George. Good success for the real Peach Congress next year. And hello, Florine.
Okay, excellent presentation. Thank you very much, Francois. It was very informative. So just to be on time, I will initiate our scientific part of the day. Our first speaker is Laura Rossini. Laura is Associate Professor in Crop Genetics at the University of Milan. She received her PhD in genetics from the University of Milan and worked in the UK as visiting scholar at AstraZeneca Plant Biotechnology section. This is really interesting. And at the University of Oxford Department of Plant Sciences. She has extensive experience in plant genetics, genomics, and molecular biology. And for the past 20 years, she and her group have applied genetics and genomics approaches to investigate the mechanisms controlling plant morphology and development. Since 2008, she's engaged in research on fruit trees, participating in the International Peach Genome Initiative and the development of genomics-based approaches for genetic dissection and improvement of fruit quality and agronomical traits. I have asked from Laura to provide us a talk with the key findings of the recently accepted research paper in plant physiology, dealing with the design and the realization of the multi-site peach collection that is considered an international research and breeding tool for fruit trees. Before giving the floor to Laura, I would like to welcome and thank Florin Stanica, who accepted to moderate this session. Florin is well known to everybody in the Peach Society. He is also Vice Rector of the University of Agronomic Sciences and Veterinary Medicine of Bucharest, member of the Academy of Agricultural and Forestry Sciences, and Vice President of the section of Horticulture. He is currently Professor of Pomology at the Faculty of Horticulture in Bucharest, expert on fruit growing technologies, including planting systems, canopy, and organic orchard management. Professor Stanica is the chairman of Peach Working Group within the International Society of Horticultural Science and the Romanian representative in the ICHS Council. I welcome both Laura and Florin, and I give the floor to Laura to initiate her talk.
Thank you, George. Can you hear me?
Very clear.
Thank you. It is a real pleasure to be participating in this event. Thank you, George, for the organization and for giving me a chance to share our initial experience with the PeachRef population. So the main focus of research in our group is on genetics and breeding of quality and agronomy traits in stone fruits, in particular peach and apricot. Clearly, this is based on the characterization of genetic resources and their valorization. And in this context, we have become interested in the application and development of phenotyping approaches for a range of different traits. Of course, we also take advantage of molecular genetic and genomic tools and approaches to combine with phenotypic data and push forward genetics and breeding. As we all know, peach comes from China, where it is deeply rooted in culture, tradition, and art, as shown in this beautiful drape representing the orchard of the peaches of immortality. But the origin of this crop is shrouded in mystery. In fact, the wild ancestor of peach is still unknown and probably extinct, but some information is starting to emerge. For example, both fossil and genomic evidence points to the origin of the fleshy peach fruit around 2.5 million years ago in southwest China. This suggests that the selection for fruit edibility started before the onset of domestication, probably involving some frugivorous primates. Regarding domestication, different hypotheses have been put forward. Initially, domestication was proposed to have started in northwest China around 4,000 to 5,000 years ago, but other evidence emerged more recently that suggests that domestication could date back to at least 7,500 years ago in the Yangtze River Valley in southern China. From China, the westward journey of peach took it first to ancient Persia, modern-day Iran, and then to the Mediterranean basin and Europe. And we have writings and paintings from the 1st century showing that Romans enjoyed peaches as we do today. From Europe, peach traveled to the Americas, initially in the 16th century, but with a second important milestone in the 19th century, with the introduction of Chinese cling to Delaware, where basically this provides a starting point for later breeding programs in the 20th century. And from the US, modern varieties arrived back in Europe and then in China, giving impulse to today's breeding programs. Innovation in orchard design, training systems, agronomic management started in Massa Lombarda in Italy, in the Romagna region, at the beginning of the 20th century. And from there, it spread worldwide, providing a first example of modern fruit culture. In parallel, advances in breeding led to the release of a plethora of new cultivars that rapidly replaced old local accessions and land races, with a consequent narrowing of the genetic basis of modern germplasm. While peach was introduced in a range of cultivation areas, humans have shaped the selection of a multitude of local cultivars, displaying an extraordinary variation in fruit shape, color, texture, and eating quality. These materials represent a cultural and genetic heritage of generations of farmers. Here, I'm just showing a gallery summarizing some milestones in US peach breeding of peaches and nectarines from the last century that provided a starting point for modern peach breeding programs worldwide. As we've heard from Maria José Aranzana in the first day of this meeting, dozens of peach cultivars are released each year, many more than in other fruit crops. But despite this, peach production is declining in important production areas such as Italy. And this may be linked to consumer dissatisfaction with the fruits they are finding on the market. What consumers are looking for are high quality fruit with excellent flavor and taste, and why not also health promoting properties. But to achieve such qualities, we need optimal combination of genetics, orchard and post-harvest management, requiring a rethinking of the whole value chain. Meanwhile, regulatory requirements are pushing for more sustainable practices. For example, in the European Union, the farm to fork strategy has set ambitious targets with the reduction of pesticides, fertilizers, and an increase of up to 25% of surfaces under organic farming. Germplasm resources play a central role in addressing these goals because they harbor a rich genetic diversity that we can mine for traits and genes of interest for future breeding, including adaptive and resilience traits and quality traits. However, germplasm collections also pose very important challenges. The high costs and intrinsic risks that are associated with maintenance and propagation of living collections are probably the main issue in maintaining germplasm collections. And this led to an effort to evaluate redundancy within collections and rationalize, let's say, conservation efforts. And this led to the success of the core collection concept, which is the idea of capturing the maximum amount of genetic diversity within a limited set of accessions. And core collections have been created for a number of fruit crops, including olive, apricot, pear, and cherry. However, intrinsic to the same concept, there's also the risk of leaving something behind and losing some materials. And this is also linked to the difficulties in sharing materials between different collections, which has led to the loss of unique materials. Finally, the fact that some materials are unique to some collections and there's not so good connections between different collections leads to problems in comparing and integrating information from different collections, where data are collected under different environments and using also different protocols. Of course, genomics can help to some extent. So with high throughput genotyping and sequencing technologies, we have seen a revolution in our ability to study genetic diversity. These technologies find direct applications in the characterization of germplasm collections, also allowing to rationalize conservation efforts. Meanwhile, these tools have opened unprecedented opportunity to study peach evolution and diversification. And finally, as we all know, DNA markers are routinely applied to dissect the genetic control of traits through linkage and association mapping. And here we have some examples. For example, in our group, we use the genome-wide association studies to dissect different traits. Here is a recently published paper on blooming data. And the use of these polymorphism data is now facilitated by databases like this one or the GDR that make it easy to find all this information and exploit it in your research and breeding program. So this is all very good, but we're still left with some important challenges. The key area that is still largely understudied is the way that genotypes interact with the environment and with management practices. And this is absolutely fundamental to really address the key challenges we are facing. If you think, for example, of vegetative and reproductive phenology, chilling and heat requirements are very sensitive to the environment, and these are changing. The environment is changing with climate change, for example. We've heard yesterday about brown rot and other biotic stresses and how influenced they are by environmental conditions. Clearly, this also applies to abiotic stress that is, by definition, dictated by local conditions. And not to forget fruit quality, where it is well known that environment and management have strong effects in creating the quality, the final fruit quality. Think sugar and acids, volatile organic compounds, but also shape. So how can these type of interactions be dissected? Since years, this has been a major topic for research in annual crops, where multi-environment trials are used to disentangle genotype, environment, and management components of trait variation and their interactions. In fruit trees, multi-environment trials require multi-site collections. The merits of these multi-site collections are several, both in terms of short-term objectives and more medium to long-term objectives. In the short term, these multi-site collections can be exploited to provide a safety net for the preservation of rare materials, creating duplications under different environments and different institutions. In addition, they can also be used as a network for variety testing to assess the performance of materials under different environmental and management conditions. In the more medium to long term, from a breeding point of view, such designs allow to overcome the usual limitations of most breeding programs, where the breeder works in a certain geographical area and selection is focused on adaptation to those specific conditions. With these designs, we are in the position to analyze genotype by environment by management interaction and identify superior genotypes and favorable combinations of genetic factors to breed improved varieties for defined environment and management conditions. Not to forget that these designs can also be used to study and better understand plant responses to external conditions at also the physiological and epigenetic level. The Fruit Breedomics project, coordinated by Francois Laurens, who opened today's session, offered for the first time the conditions to build such resources. Involving 28 research institutions and companies, the Fruit Breedomics project was an EU-funded project aiming at bridging the gap between genomics and breeding. One of the core aims of the project was the extensive characterization of European genetic resources of both peach and apple, and integrated analysis across collection. So towards the end of the project, the idea and the opportunity came to build two multi-site reference populations, one for apple and one for peach. So this is how the PeachRefPop, the peach reference population, was born. It is a unique resource to study genotype by environment by management interactions, and also providing a heritage of peach cultivation and breeding. It is present in 4 complete copies and 1 partial copy. So 2 copies are present in Spain, one in Gimenells in the Catalonia region, and one in Mula in the Murcia region in the south of Spain. 2 copies in Italy, a full copy in Imola in Romagna and a partial copy in Rome, and another copy in Naussa in Greece. The design of the PeachRefPop was a stepwise process where we had to go through a series of choices, let's say. So the first choice was to define the domain of the PeachRefPop, and the starting point obviously was the fruit breedomics project, where we had a range of materials, where we decided to focus on Prunus persica and Ferganensis. So the starting point were over 1,200 accessions of peach and over 1,400 progenies from 18 different crosses. Then this was too much material to include, be included in the RefPop, so we had to define the size, and this had to be a compromise between statistical power to run the kind of studies we had in mind and the feasibility of maintaining these materials in different orchards, in different countries, and in different institutions. So we aim for around 400 entries with a minimum of 150 accessions and a maximum of 250 seedlings. But probably the most critical point in the whole process was this step 3 with the selection criteria. Here, it was obvious to apply some of the standard methods for core collection construction, so maximizing genetic diversity and phenotypic diversity. But we also wanted to leverage expert knowledge to be sure to include some important materials in the history of peach cultivation and breeding, and always keeping a close eye on priority traits. So this was really the most interesting and complex step to some extent. And finally, the final choice of entries was as a consequence of the first 3 steps, and the allocation, distribution of the materials was also a very difficult step, but very interesting. So here you see some of these choices that we made were based, for example, on the germplasm collection from the Fruit Breedomics project. These were subjected to a series of analyses with some softwares to design core collections, integration of expert knowledge, and led to the selection of 169 accessions representing genetic diversity of peach germplasm preserved in European collections. Population structure analysis and estimates of genetic diversity were used to compare the starting fruit breedomics panel to the final set of PeachRefPop accessions that we call PRPX. And in agreement with previous studies, in both panels, we detected 3 main groups, including Occidental breeding materials, Occidental traditional materials, and Oriental materials. A number of indexes were used to check the correspondence of diversity between the 2 sets, and they largely overlap, so this was satisfying. And also we were happy with the final result representing the phenotypic diversity for a number of key traits, from maturity date to fruit weight to quality traits of the fruit like soluble solid content, texture, acidity, color, etc. Complementing the choice of accessions, also 234 seedlings from 15 progenies and the corresponding parents were included. And these were selected to represent different breeding programs in the European Union, and also to taking into account the kind of traits that were segregated in the different crosses. So altogether, a total of 403 entries composes the PeachRefPop, and they were all propagated by the same nursery by grafting on the same rootstock and distributed to the respective partners in 2015. And so they went into fields in the different locations. Also, the choice of locations was reflected some important decisions. The PeachRefPop locations are representative of Mediterranean growing areas and environments. We have a range of latitudes from 38 to 44 degrees north, a range of, let's say, temperature, climates, so semi-arid climate in Mula, in southern Spain, up to more subcontinental climates in Imola and in Nausa, where we also have more rain. And of course, also a range in, for example, chilling hours that go from over 1700 in Nausa to around 700 in Mula. Another critical point was the actual layout of these fields. The fields comprise two major blocks, the M1 block and the M2 block. The M1 block has a full set of all the 403 accessions. The M2 block has only half of the accessions. They are both present in both blocks. We have two replicates and additional replicates from some Czech cultivars. The partial replication in the M2 block is designed in such a way that between any two given sites, you have a good amount of shared materials. So where do we want to go with this kind of resource? What is the objective of building something so complex? Well, obviously, the idea, as I said, is to disentangle genotype by environment, by management interactions. And for this reason, we became interested in genome-wide prediction approaches that have been developed in humans and livestock genetics to predict complex traits based on genome-wide marker information. In recent years, genomic selection or genomic prediction has also gained momentum in plants and fruit trees. And here I try to summarize the basic principle. You start from a reference population that is both phenotyped and genotyped with dense markers across the genome, and using a range of statistical approaches like linear models or machine learning, associations are built between the SNPs and the markers and the phenotypes to build the prediction equations. These equations can then be used to characterize, let's say, another set, which could be your breeding population. You just need to genotype the breeding population, then apply the prediction equations to the genotypic information to predict the phenotypic behavior of your breeding population. And this leads as an output to so-called genomic estimated breeding values that are basically predicted phenotypes. You can include also a validation step where you actually really phenotype part of your breeding population, and you estimate the predictive ability of the model by comparing the predicted phenotypes and the measured phenotypes. So as an example of how this can work, we can look at the work that was done by our colleagues on the apple RefPop. This is a work that was published last year in Horticultural Research. The apple RefPop includes over 500 entries and was planted in 6 different European countries. And here, as a first pilot study, they decided to try to apply genome-wide prediction to predict some phenology traits. So I'm just showing one of the figures of their paper where they show that their predictive ability for harvest date is around 75%, and for floral emergence is around 57%. So these are quite encouraging results from this first pilot study. Of course, much more work will come on the apple RefPop, and I guess we will see more results from that consortium. With respect to the PeachRefPop, in this slide, I would like to give you an idea of what we are currently doing in the context of the Prima Free Climb project involving various partners in the Mediterranean. In terms of the PeachRefPop, 3 sites are involved, Imola, Nausa, and Gimenells. And we are conducting a G by E by M study of peach scion tolerance to drought stress. So here you see how we are using the design in this case. There are these two blocks that are irrigated and these two yellow blocks that are rain-fed. And the number of data are collected throughout the growing season. For example, soil water content, according also to precipitation. We identify some moments during the growing season where the rain-fed block really shows a significant reduction in soil water content compared to the irrigated blocks. And then as measurements of the plant response to the water availability, we measure tree trunk cross-sectional area and see some different responses according to genotype, and also leaf temperature derived from thermal images, as shown here, to then derive crop water stress index. And this is just preliminary data from the first year and from just one location. But what we can see is that some trees experience little water stress and some trees experience more water stress. So of course, we have to still do a lot of work to integrate data from different locations and see how this can be interpreted. Another study we started is a study on reproductive phenology, maturity date and blooming date that is also ongoing. So I would like to wrap up with some take-home messages and perspectives. What we've learned so far by this adventure of the RefPop. Well, first of all, the RefPops represent the first attempt to coordinate transnational scientific research in fruit trees to work on G by E by M of complex traits. So this is was a challenge, and we are trying to face it in this way. But other opportunities may also be considered. For example, multi-site progeny evaluation through seed exchange, which is another way of sharing materials, even if not vegetatively propagated. We're also learning about some challenges. For example, to make it possible to integrate data across different sites, we need to standardize orchard management practices, think pruning, thinning, fertilization, and so on. But most importantly, we need reliable approaches for phenotyping of a large number of trees. And for many of the most important traits, these are completely lacking. They still need to be implemented or developed, particularly for traits related to biotic and abiotic stresses. And of course, there are new methods coming along, such as based on remote sensing, but we need to assess whether these can be applied in a high throughput manner and with the necessary accuracy to evaluate genotypic differences. Okay, I would like to wrap up by acknowledging the many people that are involved in this highly collaborative work on the RefPop. First of all, the leader of our group, Daniele Bassi, who also led the establishment of the PeachRefPop, Marco Cirilli, that joined the effort and jumped into it with all his energy in the last years, and other people in our team, Stefano Foschi in Imola, who actually manages the orchard, and all the partners that have provided genetic materials to the RefPop, from CREA, from IRTA, from INRA, and also for the partners that host a copy of the PeachRefPop in IMIDA, in Greece, and other people that helped us to characterize different locations, design the layout of the fields, and importantly, last but not least, help with the phytosanitary measures when we had to share the materials. Thank you very much for your attention, and I'll be happy to answer any questions.
Thank you, Laura. A great presentation for a great job. I just received here some appreciation and questions also. One is from Francois Laurens, who asked me to congratulate you for the great job that you did and for your initiative in fruit breedomics, and the fact that actually you follow up the PeachRefPop job and you are continuing to do it. And also a question from him. The idea is to have a link between apple and peach he reminds us that the Apple RefPop is running now thanks to the European fundings in the frame of the Invite project. So the idea is to invite you to work together again. So certainly fruit breedomics was a great project that allowed to all the geneticists, breeders, and pomologists to exchange their knowledge and to work together in Europe not only. So how do you see this possible link again between RefPop in Peach and let's say Invite project in Apple?
We strongly believe in building capacity across different crops. In fact, even in Free Climb, the project coordinated by Daniele Basti, in which we are supporting the current work on the RefPop, we are trying to establish new reference population, also for apricots. So we strongly believe in building more of these resources and obviously sharing experiences, challenges, and why not also lesson learned, but also some good practices that can help in best managing these resources, which are very difficult to maintain, but I think also offer very, very nice opportunities. So thank you, Francois.
We have 174 participants in this moment, and please use the Q&A button to ask questions. And I have already received several here. And one is about, is there any impact in drought condition and stress condition intolerance and blossom of fruit and fruit?
Okay. Obviously, when you apply these differences in water management, there could be many traits that we could measure. Let's say in the first years, we focused on the traits I mentioned, but of course, the idea is also to start collecting more data. At the moment, I cannot comment, I cannot give results in response to this question, but yes, the idea is with the efforts that we can make to collect more data also on fruit, for example.
Okay, there is another very short question. If have you identified any markers related to early ripening peaches from your study?
From the PeachRefPop or from?
Yes, from the PeachRefPop.
No, we haven't done, as I said, we have not elaborated all the data yet, and we were focusing on these indexes that I showed, but with more data, we will be able also to answer this question.
Okay, I would like to propose you to leave some questions also for the last part of our session today.
Sure, it will be a pleasure. I will also check the question and answer.
Yeah, okay, and please also try to answer some of the questions that are on our chat. Thank you so much, Laura, once again. Mille grazie. So I propose to pass to the next breeder. So George, the floor is yours.
Thank you. Thank you, Florian. Thank you, Laura.
Thank you.
Next speaker is Nicos Papadopoulos. Nicos is a full professor of applied entomology and the director of the Laboratory of Entomology and Agricultural Zoology at the University of Thessaly, that's located in the city of Volos in Greece. He completed a PhD in entomology at the Aristotle University of Thessaloniki, and then followed postdoctoral training in the Department of Entomology at the University of California at Davis, focusing on insect biodemography and on behavioral patterns of insect aging. His research interests are focusing on the ecology, evolution, behavior, and management of insects, particularly that of fruit flies that are considered the most important group of invasive pests. Understanding local adaptation and the interplay between natural selection, gene flow, and plasticity in major life history traits is the main venue of his current and future research. Nikos' talk deals with the description of invasive pests that threaten the sustainability of peach fruit production. Nikos, thank you very much for joining us, and we are looking forward to your talk.
Thank you very much, George. First, I would like to thank you for the invitation. I'm really happy to participate in this event that is really successful and highly attended. So my presentation, as you said, George, is tackling a very interesting and important aspect in plant health and plant protection. That is the biological invasions, and apparently focuses on the peach production. So this is an outline of my talk. First, I would try to define a little bit the biological invasions and point to the important aspects that should be considered for the management. Then there is a reference to peach production, especially to highlight the zones of the geographic zones of of peach production in order to define the movement of the different pests. The introductory part of my talk also involves the presentation of major pests with some focus on some Coleoptera, some beetles, some Hemiptera, Lepidoptera, and then of all this long list of pests, I focus on fruit flies, which is in fact the focus of my research for the last 25 years. And I will explain you why fruit flies are such an important group of pests and why it is considered also a model group for the invasion biology. Then I will present you a new project. It's actually running for the last 18 months. It's a European funded project that focuses on invasive fruit flies that threaten the pome and Prunus production in Europe. And of course, I have some ideas for a take-home message at the end. So going to biological invasions in the simpler way, they are described as the expansion of the geographic distribution of a species into an area that that was not previously occupied by this pest. And there are actually two processes there. The one is the range expansion. It's a gradual process that involves movement of the pest out of its native or current, let's say, distribution, but it's without any human assistance. It's based on the dispersion, on the flight ability, on the movement, short distance movement. This is a gradual process and needs a specific way of management. On the other hand, we have more dramatic invasion events. I call them as long jumps, and these are mainly assisted by humans, by human activity, trading, and movement. And these long jumps involve movement of organisms across borders and across continents, from one continent to the other. So such major invasion events can be dramatic for the invaded area and can result in huge economic losses, but also can result in major social aspects in these invaded areas. The phases of invasion, as are outlined here, it first is the arrival of an organism into the novel environment. Then it's this establishment, which is a self-sustained breeding of the population and the ability of the population of the new pest to overcome all the biotic and the abiotic stresses of the new area. Then we have this phase of naturalization. It's another step. Sometimes it's under the establishment step. And this naturalization involves all the interaction of the new organisms into the novel environment and how this organism is integrated into this new environment. After the conclusion of this naturalization phase, which cannot be predicted and can last from a few years to several decades in the different organisms, we have the spread, the expansion of the distribution into the novel area. So all these steps are associated with specific management and intervention practices. So what are the key drivers of biological invasions? First is the climate change, the global warming, or I prefer the climate change. And this is because previously hostile environments, because of the warming or the climate change, they became favorable for the breeding and the performance of novel organisms. The second driver is this massive trading, massive food trading, because of the globalization. And we have in this fruit trading, we have massive amount of fruits, fresh fruits, that can export it from one area, from one continent to the other. And apparently this trading brings also harmful organisms. And this is a huge propagule pressure in the importing countries. This is also, let's say, facilitated or enhanced by the increased human mobility. The pre-COVID era was, I suppose, the peak of human mobility. People want traveling a lot, and often they are bringing some plant materials with them, and then they bring also harmful organisms. So these are the key drivers of biological invasions. And we have, in addition, changes in land use that in a way they loosen the defenses of the environment, if I can use this word. Also, we have new cropping patterns. We have new cultivars. We have a broadening of the ripening season, and all this establish a very favorable environment for the establishment of new organisms. So going now to the nectarines and peach production, you know much better all these statistics, but what I'm trying to do here is to define the two zones. In fact, most of the peach and nectarine production is taking place in the northern hemisphere, and it seems it's this zone of the production that has southern and northern limits. And also there is another zone in the southern hemisphere. So the harmful organisms and pests can move from south to the north, and from east to west, and from west to east. So we have a really complex environment, and with all this trading and movement of fresh fruits and vegetables, we have really a very complicated environment with new organisms arriving and threatening the production of nectarines and peaches. So, looking at the list of pests, of peach pests, we have really many coleoptera, many beetles that can come to our orchard and harm the peaches. Then we have bugs. We have a Miptera that can also infest the peaches, and we have Lepidoptera. The family Torticidae is the most known, and we have a list of pests there that can infest the foliage, that can infest the fruit. And last but not least is this group of Diptera, the Diptera tephritide. And you see, only in one family we have a very long list that can infest peaches. And we have also major pests for peaches in this category. So apparently I cannot cover in this talk all these pests. And what I did is to select and present you one beetle that is currently expanding its distribution in Europe. So it's a specific example of an invasive pest. Then going to a pentatomid and present you another organism that arrived in Europe and in North America the last 2 decades, and soon it became the major pest or expanded its distribution all over the different countries. Then I have a Lepidoptera species that is not currently in Europe, but it's one of the major threats, not only for the peach production, but for the production of citrus and of other crops. And the focus then is on the fruit flies, and I have a group of fruit flies that I will elaborate a little bit more. So we may have pests that infest the wood, the trunks, or the shoots of the peach tree, pests that can infest the leaves, and pests that can infest the fruits. And starting from the wood, we have this Aromia bungi that has been detected in Europe. It comes actually from this area, from the Oriental area, and then it arrived in Europe. It seems first detected in UK, and then in Germany and in Italy. And this is a pest that can kill the tree. You see that it's a good boring insect, and it's one of the important concerns for the peach production in Europe. Because of its biology, as we can see here, all the Cerambicids, they have a really long life cycle. We have larvae in the trunk of the tree for years, for up to 3 years. They overwinter there. They continue their feeding and they harm the tree, but it's hard to be detected. And this is a major issue. On the other hand, this habit and long life cycle prohibits the aggressive dispersion of these pests. But this is a specific example that currently is posing some issues in the European horticulture. Then if you go to another pest, we go to pests that can harm the fruit, but also, give me a moment, but also the leaves. It's the brown marmorated stink bug. And I brought this example because if you see at this map now, it's all over the Northern Hemisphere in all this zone of peach production. And but if we look a little bit at the patterns of invasion, again, it comes from the eastern parts of Asia. And in 1996, it was first detected in the United States. Then in 2007, it was the first detection in Europe, in Switzerland. And since then, in very short time, it invaded all the European countries, and now it's all over the Northern Hemisphere. And recently, also, we have seen it in the Southern Hemisphere in Chile. So this is a very aggressive pest. Actually, this time of the season, we are receiving a lot of calls in my laboratory, reporting high populations of this pest in the peach producing area of central Macedonia in northern Greece. So the pest can harm, as you can see here, the fruit. And I wouldn't put it now in the high priority of invasive pests because it's all over and now it's under an integrated pest management control. But this is a very good example of how fast this invasion can proceed. So focusing a little bit more on the fruits now, we have the Thaumatotibia leucothreta, the false Cuddling moth that is native to sub-Saharan Africa, but also recently has invaded Middle East. And this is considered a very important pest, as I said before, not only for peaches, but for other fruits as well. What is important in these pests of the fruit is that they can escape detection, they can escape interception, and they can arrive with cargo shipment with fresh fruits that can that are exported from South Africa, for example, to Europe, that are coming from the different parts of the globe. So there is a huge propagule pressure, and it's often very difficult to detect and to see the infestation. This is the case with this pest. This is a multiple times pest. It's an aggressive pest, and I hope we prohibit its entrance into the European continent. And now I'm coming to the main part of my talk, which is fruit flies. And why fruit flies are important pests and important as a model organisms in invasion biology as well. This is because they have a rather cryptic life cycle. A big part of their life cycle is within the fruit. They come to the fruit and they infest the fruits when very close to harvest. This oviposition things and the oviposition and the infestation cannot be detected at this point. So they can easily escape any notice, and they can often arrive into new areas with this importation of the fresh fruits without being noticed. So we need a really elaborated strategy in order to deal with these pests. So the eggs are deposited into the plant tissue. We have this cryptic behavior. Then the larvae destroy the fruit and leaves the fruit to pupate in the ground. Sometimes fruit flies can move from one place to the other with root stalks or with plant materials. But the main pathway of entrance into Europe and in other places, it's through infested fruits and through cargo shipment with fruits that passengers can bring with them. So the importance of this group of pests is highlighted by a long debate about the invasion in Northern America mainly and the establishment. And this regards medfly. This is a never-ending, I would say, debate with the medfly invasion in the States and then the invasion of additional pests in California. I was involved in this debate. It's like there are groups that argue about the establishment of these pests in California. There are other groups that argue against this opinion, and this, as you can imagine, creates a friction. And the debate is going on because of the importance of this pest and the estimates of establishment, for example, of this pest in California. They give a really huge economic burden for the growers and the fruit trading industry in this place. The same apparently holds for Europe. But in Europe, we have already some species. We have the Mediterranean fruit fly. But looking at the interceptions in Europe, we see that a big part of these pests are intercepted every year. Fruit flies are the most frequently intercepted harmful organisms in the European ports of entry. It seems that more than 15%, 16, 70%, up to 20% of all interceptions are fruit flies. If we focus on the fruits and vegetables, this proportion increases to 30%. 1/3 of all interceptions of harmful organisms in fruit and vegetables are fruit flies. So we have a very huge probable pressure of this pest in Europe. Most of them because we can detect infested fruits and we have larvae in infested fruits, they cannot be identified to the species level. So we need to develop tools in order to identify all the specimens in the interceptions. And in fact, here you see that only a small proportion of the intercepted fruit flies can be identified to the species level. Because of, if we try to focus in some species of this group, we have, as I said before, the Mediterranean fruit fly. Why I consider it as an important invasive pest. It's a pest that expands its distribution in Europe. Traditionally, this pest is established and causes problems in the coastal area of the Mediterranean countries. It is considered a major pest of citrus, but in the recent decades, we have detections and infestations in Central European countries. We have problems with medfly in northern Italy. We have problems with medfly in northern Greece that 20 years ago, there were no any issue, and we have this expansion of the distribution. So medfly is moving to the peach producing zone, and gradually it becomes a dominant pest in this zone. Then we have two additional pests. The one is the Bactrocera dorsalis and Bactrocera zonata. The Bactrocera zonata is called the peach fruit fly. And why these are important? Because the first one is a really aggressive invasive species that is all over the African continent below Sahara, southern of Sahara. and it arrives in Europe quite frequently. And the other pest is already in Northern Africa and in the Middle East, the peach fruit fly. But it's not only there. Now we have detections of these invasive fruit flies in Europe. We have detection of both the peach fruit fly and the oriental fruit fly, Bactroceras zonata and Bactroceras dorsalis. In Austria, if you can see here, for several years we have detection of the peach fruit fly in Austria, but Austria is considered as a hostile environment, that there was no much, let's say, stress on this pest. But a couple of years ago, we had the first detection in Italy in a friendly area of Bactroceras dorsalis. Then we had a detection in France. And this rings the bell that these flies are arriving and they escape all these interception measures, and they have breeding populations. They are considered currently as transient populations, not established, but it's like a really important threat for Europe. If we look at the medfly and the range expansion of medfly, as I said before, we have this emerging as a pest in the most temperate areas in the peach production zone, in all major peach producing countries like Italy, Spain, Greece, and also we have detections in other European countries like in Romania, in Bulgaria, we have in Austria, we have in Germany, in Switzerland. So there is something going on, and this new map of EPO actually includes all these countries as countries with established populations of medfly. And this range expansion, apparently, it threatens new crops like the palm fruits, the apples here, the peaches, and threatens also or puts some stress on the successful integrated pest management programs, such as the one of apples and peaches in these areas, because these integrated pest management projects have addressed in a way the Lepidoptera pests. But now you have a new pest that comes very close to harvest. So you need additional application of pesticides. And this, in fact, destroys all the IPM scheme in this area. So to summarize it, fruit flies are arriving in Europe in high rates. So we have a high propagule pressure. We start having detections of new tropical fruit flies. Medfly emerges as a new pest in deciduous fruit orchards. And in addition, there is gap in ecological and climatic modeling in Europe about, with focus in Europe about these pests. And we do not have a very well coordinated interception and detection system, and we lack fast and aggressive responses to eradicate and contain these flies. So dealing with biological invasions, we have these phases this is the timing of the invasion. So we have a phase that is the prevention, and we have specific measures that should be applied in the prevention phase. Then we have the early detection, and then we have the management and the eradication and the containment. If you see at these triangles, the earlier the intervention, the lower the cost, and the higher the management efficiency. If we go down to the invasion process, the management cost is really high, and the success is really low. So to deal with biological invasions, we should focus on early responses. We should develop risk assessment and pathway analysis, predictive modeling. Also, we should work on prevention, on updating the legislation, on developing more efficient quarantine treatments and interception strategies. Also on updating the detection schemes and detection strategy. And then, of course, we have this eradication and containment that, as I said earlier, they are not often that efficient. So that's why we established a consortium, and we have a European funded project. The project is Fruit Flies IPM in silico boosted pest prevention and management of fruit flies. And the vision of this project is to protect the European horticultural trade from the current and imminent threats posed by selected emerging and new frugivorous fruit flies. And the focus of the project is these three pests that I mentioned earlier, on medfly, on the peach fly, and on the oriental fruit fly. This is the approach of this project. We've seen the seasonal biology of medfly in these cooler areas as a parallel to the invasion process of the other two pests. In fact, the time scale is different because we have a seasonal biology on the one side, on the left-hand side, and we have the invasion process on the right-hand side. But as you can see, we have long winters and spring that we cannot detect the population here. This is the population density. And then all of a sudden, in the mid of the summer, we have a boom of the population. We have an increase of the population. So the target here is to bring these medfly populations under the economic injury level in order to achieve this IPM approach. On the other hand, in the invasion process, we have these unknown parts of introduction and establishment, and then we have detection that can be considered as a transient population or detection that may stem from an established population. But again, the process is the same. If we detect early these flies, then we can have an eradication intervention. Otherwise, we should go for an IPM project. And if we fail in both cases, we lose most of our crop. So the FFIPM project focuses on prevention and develops tools for interception, early detection, and also off-season management for medfly. And we invest on spatial modeling and on generating new knowledge and developing different tools. Some of these tools I will show you in the next few slides. And then at the end, we develop an IPM scheme. So we are refreshing this predictive ecological modeling, and we try to understand the climatic suitability of the different areas of Europe, of the areas that peaches are cultivated. And we have a group from Australia, led by Darren Kritikos, who is updating the climax modeling, and we would like to have real-time predictions on the distribution and the suitability of the environment. So these are some maps for medfly, new maps that have been established and developed by the FFIPM project. This is for the medfly, and this is for the oriental fruit fly. And if we focus here in Europe, we can have different scenarios regarding the suitability of the habitat. So this is one part of the work. On the other hand, we are working on developing interception and detection tools. We have a very good group in the Benaki Institute in Athens, working with some Italian colleagues, and they try to develop a system, an in-know system, to detect infested fruits in cargo shipments. This is really important for the interception of these pests. And the earlier the interception, if we manage to intercept the fruits, then we can prevent the introduction of these pests. Also, we have a nice group in Israel working, and we're working together on developing e-traps, and a group in Italy working on the same concept. And we have already prototypes of these traps, and these traps will be included in our experimentation of this year. In addition, we work on developing morphological keys and molecular cues to identify to the species level the different intercepted and detected species. Moreover, we are going from the continent, from the European continent, to specific areas. One of the target areas is Croatia. The other area is northern Greece, the area of Central Macedonia, and we try to develop spatio-temporal modeling in order to follow and detect earlier the populations of medfly in these areas. So this is based on the Timex modeling that uses data on development of fruit flies in the fruit, in the development in the soil, and also aspects of the canopy of the tree. So to wrap up, and this is a kind of a take-home message, the biological invasions, and especially the fruit fly invasions, need a holistic approach. We need to increase coordination among countries in Europe. We need to develop trans-border actions to address all steps of invasion, to invest in generating new knowledge, And also we need to develop databases and web-based repositories in order to share our data. Also, we need to revisit and think of aggressive response strategies, eradication and containment campaigns that will be adapted to European conditions, because such strategies exist for other parts of the globe, for Australia, for Southern America, from Northern America, but not for Europe. And also we need to invest on training and involvement of all stakeholders across the trading chain. So this is to acknowledge the partners of the FFIPM project. There are 21 partners, more than 70 scientists, and also to acknowledge the contribution of my student Vasilis Rodovitis, who helped me in preparing this talk. So thank you very much, and I will be happy to answer some questions. Thank you.
Thank you, Nikos. Great presentation. A big problem with the invasive pests, with the globalization. Everything is traveling north, south, east, west, from all the continents to different conditions. Pests are already always adapting to the new climates, and as you mentioned, we need to be prepared, and we need to find the best solutions to the containment of these pests and then to fight them. So please use the Q&A button for questions. Just by bad chance, the Ceratitis capitata was first time found in Romania in one of our orchards in the university. And as you mentioned, even we are in the colder climate, this fruit fly found a way to live here and to multiply. So we need to take care.
I would like also to stress, Florin, the fact that for the peach society, there is a lack of experts dealing with pest management. So Nikos used to work with Rangoletis in sweet cherry. So it's very important that he has secured funding. So through this EU-funded project, he can work also on peaches, because I think that we do not have a critical mass of researchers about peach and pests. And Nikos, you can elaborate on that if you want.
Definitely, George. Actually, there is a focus, as I said, in this project on the peach production. And we have spotted several areas in Europe. The one is Central Macedonia, the big peach producing area in northern Greece, but also the area of Rome in Italy. We have Italian partners that we are collaborating, and we try to address the problem of fruit flies for the peaches.
Nikos informed me that a big group of researchers and scientists who are being involved in this EU-funded project will join us in NAUSA next year. So I think they will exchange ideas with peach experts about tree physiology. So maybe we can increase and raise more awareness about the importance of pest management on peach fruit. If there are any questions for Nikos, you have the opportunity to ask, and Nikos will have the time to reply to them during the concluding remarks. So in order to be on time, I would like to introduce our third speaker. Nikos, thank you very much for your talk. So our third speaker is Tom Gradziel. Tom is professor at the University of California, Davis. He completed his bachelor degree in plant science at the University of Massachusetts, followed by a master and doctorate degree in plant breeding at Cornell University. He is responsible for the genetic improvement of almond and processing clingstone peaches. Among others, Tom is chair of the International Society of Horticultural Sciences Almond Working Group and member of the Rosace Genomics, Genetics and Breeding Executive Committee. Breeding methods employed in his program include wild germplasm characterization, preservation and introgression, epigenetic selection, intra and interspecific hybridization, and marker-assisted breeding. The breeding program has released 8 peach varieties, 3 almond varieties, and 1 rootstock. The title of his talk is very important, especially for the peach canning industry, because it deals with challenges and opportunities on breeding peaches that are being destined for processing. Tom, good morning in California, and just to let them know to all attendees that Tom will additionally act as keynote speaker in person next year in Greece.
Thanks, George. Glad to be here. Actually, I'd be happier to be in Greece as this was originally scheduled. So, as George mentioned, I'll be talking about the processing peach, the poor cousin to fresh market peach. Poor because the lower value of the crop. And because the lower value of the crop, we need to both maximize yields and also maximize orchard longevity. Typically, we're looking for an orchard productive life cycle of about 20 years, and we're looking for yields of about 20 tons per acre, which would be about 45 metric tons per hectare. And right away, this changes the breeding program. We've talked a lot about how genomics and molecular markers can improve the efficiency and the speed of our ability to generate new cultivars or generate new candidate cultivars. But when you have the crucial or the remaining part of the breeding program is the extensive testing of these varieties in the different growing regions and for the different orchard life expectancies. When you have a processing peach orchard where that grower will either make or lose money depending on that last decade of production, the only way that you can have a good prediction that it'll be a successful variety is you need to test these varieties over that 20-year expected lifespan. And right away, you've got a much higher life cycle turnaround, a much longer life cycle turnaround in the breeding program. And also because of the lower cost, we need to minimize, because of the lower value, we need to minimize costs. We've talked, many of the speakers have talked about how increases in regulations, increases in chemicals, and particularly increases in labor are some of the major challenges facing both the fresh market and the peach industry. Another unique aspect of processing peaches is because of the bulk harvesting, the bulk transport, the rigors of the processing process, we need a peach that's much more tolerant to physical damage. And because of that tolerance, almost all processing or all canning uses the non-melting clingstone. We do have some processing in terms of freezing, in terms of purees and juices, for instance, that uses the freestone, even some canning, but the great majority is with processing clingstone, the non-melting clingstone. And the trait we're particularly looking for is that non-melting trait. The non-melting gives us the firmness so it can tolerate the physical abuse in harvest, in transport, in processing, which is highly desired. The clingstone aspect increases the likelihood of having pit fragmentation, particularly when we have the mechanical pitting. So as that flesh is ripped apart, it can sometimes take fragments of that pit ridge. And this is a major problem with the canning industry in terms of the consumer problems. It's not desired, but as we'll see later in the talk, it is required in order to get those characteristics that we're looking for. So in many ways, processing peach, because of its bulk handling, not only bulk harvest, but bulk management, is more similar to the other crop I work with, which is almonds. And so for the talk, I'm going to be talking about using almond genes and some of the almond approaches to improve cultivars. Basically, the idea of new genes for new and even traditional problems that we have within the industry. The idea of using almond for peach improvement, and that includes not only the domesticated almond, but the wild almond species, of which there's anywhere from 20 to 40 species, isn't that outlandish. Basically, almond and peach are readily crossable. There's minimal crossing barriers. You can consider them almost the same species, almost the same genome. The exception is that peach has evolved, as Laura had mentioned, in South China. The slide here shows this is near Xi'an, where some of the more recent research indicated that maybe a center of origin for peach is going into the Gansu province. But it shows that this environment is almost subtropical. It's a fairly mild, warm climate, as opposed to almond production, or the almond and its wild species, which originated in Central Asia, typically around the Zagros Mountains in Persia. This image here shows it's on my way to Isfahan through a mountain pass through the Zagros Mountains. We're at about 9,000 feet. And these are the wild almond species, Prunus Scoparia. It's the only plant that can survive at this very arid, very high altitude. It's a bush, very small leaves, fruit, and the flowers, small leaves develop in the spring, develop into the fruit. But as soon as the heat comes, the leaves basically drop off the plant, and all the photosynthesis is on these green stems, almost more a cactus than an almond. So that same genome within almond has evolved to a genetic tool chest very different from that of peach. And it allows us a lot more options in terms of with emerging production problems. So my talk is going to be emphasizing using some of this germplasm, and I'll include both domesticated almond, some of the wild almonds, as well as some of the wild peaches and some of the heirloom peaches, towards targeting 4 major goals we have with mechanical harvesting peaches. One is a once over harvest, the ability to go in once with our mechanical shaker, or even with hand labor, to get all the fruit off the tree and not have to revisit the tree. Second, inherent with this type of mechanical harvest is damage and the increase of disease. And so we need to improve our brown rot resistance. And finally, we need to modify both the fruit architecture and particularly the tree architecture to make it more amenable, not just to mechanical harvest, but things like mechanical pruning, mechanical flower thinning. This slide is a busy summary of the breeding program, showing the different crosses and to develop some of our major parents at this time. But it shows our major sources. In addition to the traditional germplasm, we've used heirloom peaches, particularly some of the things from South Africa have been very useful to us. And we suspect these were originally brought to South Africa from India. So things like the Walgant and the Voltamate peach. The subtropical peaches, Maria Zenia had talked earlier about the Bolinas, which was developed by Dr. Nakasu and Pelotas and Embrapa, developed from some of the land races there, which have a very good tolerance, very good ability to produce fruit under high brown rot, high rainfall conditions. The wild almonds, and again, we're looking at about 40 different species. Typically, those most readily crossable to peach include about 12 to 15 species. The cultivated almond and its wild relative, probably Prunus fenzliana. And finally, some of the wild peaches, particularly Prunus myra and Prunus davidiana. Show some of the again, peach and almond are very similar. They're similar in the general layout of the genes on the chromosome. And this means that when we go into meiosis in terms of pollen and ovule development, that we have successful meiosis, successful development of the gametes. We don't have a high lethality because these chromosomes recognize each other. They're able to attach to each other, have successful crossing over, and have complete chromosome numbers within the gametes. The other thing that this slide shows is that we do have some recombination. We have some evidence that we have things such as translocations or inversions. So the structure of the chromosome, it's largely the same, that we have these recombination, so we can bring almond genes into peach, peach genes into almonds. But we also have a number of exotic changes, both the chromosomes into new segments, into new arrangements or relationships with nearby genes. And this gives us the opportunity to get perhaps novel recombinants and perhaps novel traits. A typical breeding strategy used by our program, the image at the upper right shows some of the recent work we've done with the agaki in Japan, showing the domestication patterns of the evolution of the domesticated peach, the freestones, the ornamental peaches, some of the processing peaches, the land races, and the wild species, both the wild peaches and the wild almonds, showing that by including the wild species, we dramatically increased our genetic options. We dramatically increase the gene pool available to us with the breeding process. So typical breeding operation, we would cross our peach and our almond. The hybrid, again, would be something intermediate, what you would expect with peach and almond. What makes peach unique is the expansion of the mesophyll. What makes almond sort of unique is that mesophyll doesn't expand, but remains more leathery, dries down, becomes the hull. Interestingly, the hull is a very important byproduct of almond production, and that hulls are a major food for the dairy industry. Just like peach, they have a very high carbohydrate, high nutrient value, and so they're very important for the maintenance, the balancing of the diets for the dairy cows. But the typical what we would do with the hybrid is we would self the hybrid, and very quickly you see the sorting out towards a peach or an almond type. So this would be an F2. We'd see similar things with the F1. We'd see the range from everything that's a almond type to things that are more of a peach type. And it's interesting when you go back to the centers of origin. If you go to Central Asia, where again, as I say, peach and almond should be considered the same genus because they're ready crossability, and yet in these areas they would give it a completely different genus. They would give it the genus Amygdalus. And when you ask the taxonomists, the systematists, they acknowledge that the crossing barriers aren't there. But when you look at the way these genes have evolved in a very different climate, it's more suitable to consider them a different genus, even from prunus, from peach. So after the sorting out, we would back cross either to peach for those lines that we want to improve our peach breeding. We also back cross these F2s to almonds. A major project in our almond breeding program has been to transfer self-compatibility, self-fertility, self-fruitfulness from peach into almonds. And then after that, several back crosses until we get the peach type fruit and a peach type tree that we're looking for. And again, it's fairly rapid process. Once we go towards one type or the other, it very quickly moves towards a peach type. We don't see much linkage drag. We don't see much undesirable pleiotropic effects. And the end consequence is both that characteristic which we may be targeting, such as improved disease resistance. Oftentimes it's sort of traits which are unanticipated, novel traits that we can only see once we get it into the proper background. And this slide here is an example of that, and it's what we call the stay ripe trait. It's the normally within both processing and fresh market peach, the peach will ripen, and after which it'll deteriorate rather quickly. And often that's been considered to be a developmental process. What we found is that it's basically a programmed developmental process, that we could change that program. We could keep the peach from rapidly degenerating, which would be desirable in nature because it would make the seed more available to the birds and such which disseminate it, but maintain that fruit integrity for a week to 3 weeks. And this is what we call the stay ripe trait. And it's been very important to us to achieve one of our most important requirements for mechanical harvest, and that's the ability to do once over harvest. Ideally, what you would want is uniform ripening within a peach, but that proves to be very difficult. And that's difficult because you have so much variability, even from the beginning of flowering. With the change in chilling requirements, it's not unusual to see some fruit where you have some flowers where the flowers are just opening, and at the same time you have on the same branch flowers that had opened 2 weeks before and had already small drupelets. And that early development is going to be very difficult to catch up with, impossible. Then when you get the normal development within the tree, you have some fruit developing in the interior where it's shaded, or on the north side of the tree where it's shaded. And since they develop in a direct response to the heat units, it's inevitable, or it's almost impossible to get all fruit to uniform ripen. And our approach has been to, rather than to get uniform ripening, to delay the degeneration of those fruit that ripen first. So the first fruit ripening on the tree basically maintain both the structural, the flesh, as well as the nutritional integrity, the flavor integrity for a week to 2 weeks and longer, and allows us to the underripe fruit to catch up. And this trait, in addition to giving us that once over harvest, gives us a range of other improvements, which I'll talk about in the next slide. Showing an example here, and this is our compact number 2, harvested July 7th. 2 weeks later on August 4th, we still have good processing. We've got good firmness, we've got good sugar, we don't have the breakdown, we don't have the development of the alcohol, aldehydes and such, which degrade the flavor. A chart shows here in terms of the flavor. And again, I've got these in pounds, but we're looking at about 3 kilograms here, maintaining to about 2.3 on our stay ripe varieties, Vilmos and Compact 2. You compare them to the standards such as Clamped and Andros. Very quickly, a loss in integrity of that fruit, loss in firmness of that fruit, which would make them unacceptable for processing. So that stay ripe limited change, multiple benefits. And by this graph, I try to, on a typical orchard, you would have ripening somewhat of a bell curve, a standard bell curve, a standard uniform distribution. Growers aren't paid for undersized fruit, which would be 6 centimeters or smaller approximately. So when a grower is making that decision of when to go out and harvest, particularly mechanical harvest, or increasingly even with hand harvest, we can't afford multiple harvests, we'll find that time where he gets the optimal yield. But where he gets that maximum yield, he's still losing about 5% of his crop because that crop has moved to being overripe. Fruits fall and fruits become too soft to be processed. And at the same time, you have 5% or so that's underripe. By allowing these fruit to suppress that degeneration, to continue normal development, to maintain normal fruit texture, fruit quality for a week or more, it allows these undersized fruits to come up to size, to come up to maturity, to come up to high soluble solids and the high flavors. So we've not only been able to capture the whole crop in terms of the production, but we've increased both the size, the sugar content, the quality of these otherwise undersized crops in our final thing. We've also improved disease resistance because a lot of our diseases, particularly things like brown rot and such, are particularly a problem when the fruits start to degenerate, when you start to having discontinuities within the cuticle, within the wax layer and such. By maintaining that integrity of the fruit flesh, we also maintain its resistance to disease pathogens. One of the things we've been interested in is taking this stay ripe, this ability to maintain its integrity past the time of the initial tree ripe into freestones. And this particular slide shows perhaps one of the few examples of what we consider a non-melting freestone. Some of the work we did with Cameron Peace early on shows that genetically this item, and this is a peach by Almond backcrossed and selfed, it's genetically a clean stone and phenotypically a freestone, an air freestone. When I first identified this peach about 15 years ago, I was showing it to Ted DeYoung. I had the peach, bounced it on the floor a few times, full tree ripe peach, opened it up and popped the seed out as a air-free stone. Unfortunately, with these in progeny from this particular lineage, while it does appear to be a non-melting and maintain its integrity, within about a week or so, it rapidly softens, and it could be even sooner depending on the environment. So it has the characteristics, but it hasn't proven itself in field performance. And the reason for this, I think, can be understood again by comparing peach with almond. In almond, just like peach, we have that channelization within the seed, the pit, or the endocarp. With almond, it's within an intermediate layer. So we can see that channelization just as we see with peach. We also have an outer lignified layer unique to almond. And in some ways, for processing peach, it would be desirable to have this type of an outer and apricot type pit because it would reduce our fragmentation and reduce the risks to the processors. And in fact, this is not what we want. And the reason can be seen in this slide. So what you need to understand with peach is that much of that mesocarp is developed from radial rays, radiovascular strands that radiate from these main bundles, which are in the channels within the peach pit. In almonds, we have those same bundles, but as soon as we put that lignified outer layer, we reduce the opportunities for these rays to go into the mesocarp just through these pores. So the potential size of the mesocarp, the potential size of the fruit is dramatically reduced. The other important consequence of this idea is the realization that since most of our peach flesh is from these laterally radiating vascular bundles, radiating out from these channels, the integrity of that fruit is dependent upon maintaining these connections, maintaining connections to the peduncle. In a processing clingstone peach, such as shown here, and I should point out, well, that red color in the processing peach, in this particular peach, makes it maybe attractive and also showcases that radiation of the bundles that I want to showcase here. This is something very undesirable for a processing peach. And it's undesirable because the red is anthocyanins. Anthocyanins are water soluble and anthocyanins are heat labile. So when we heat process these things, when we can these things, that red pigment, the anthocyanin, will bleed out and it'll turn brown as sort of this undesirable ooze. So breeding operations have to aggressively breed against it. Also, because lignin and anthocyanin are in the same pathway in terms of lignin development, when we see these red pits, it's also an indication we have a much greater vulnerability to pit fragmentation. But the desirable thing of the clingstone is that as that peach goes into full ripe and beyond, we maintain the connections between the mother bundles connecting into the funiculus with the bundles which basically constitute the flesh. And this is shown perhaps clearer in this slide. So in this slide, I've shown fresh market, the freestone peach versus the clingstone, our freestone non-melting, and then some of our stay ripe. The graph shows pressures in a time pattern. We've got a penetrometer. And so with the freestone peach, it's freestone and melting because the freeness from the stone, and also so the endo-PGs that are causing the attachment to those, to that pit bundles upsize, and also the endopolygalactonase acts on the cement or the connections between the different strands. So these strands are no longer attached to each other, they become separated. This allows both a physical separation and more aeration within this fruit, allows the development of a lot of these secondary metabolites, these secondary flavor compounds, which makes freestone so desirable for the fresh market. For the clingstone, we don't have the abscission between these strands so that the flesh maintains its integrity, maintains its firmness, and we also maintain the connection between those bundles and the stone. In the freestone non-melting, the strands remain glued together, if you will, that we don't have the separation, we don't have the endo-PG degeneration of the middle lamella between them, but you do have the separation between these mesocarp strands and then the major feeder bundles that are attached to your stone. And so our suspicion is that by inherent within that freestone characteristics, that abscission between these strands, between the base within the feeder strands, the larger channelized feeder strands, we lose the ability to nourish that fruit once tree ripe, once that separation occurs. So they may be genetically programmed to sort of not degenerate, but they're basically starved out because of that connection, that attachment to the nutrition is lost. And our Statewide Trait is based on a clingstone, so those connections are maintained. We've reprogrammed the flesh, if you will, so it doesn't go into that programmed degeneration, and it can maintain its integrity for 2-3 weeks or more. An analogous and interesting situation is also seen with pitless fruit. So I'm showing the Luther Burbank's spent much of his career trying to develop a purely pitless plum and using some of the French cultivars as a starting point. And he was successful, except he always found this one segment of highly lignified. And then Ann Callahan's group with the USDA years ago showed that it was possible to totally shut down the lignification of the endocarp. But the problem here is this tissue isn't part of your endocarp. This is part of your funiculus. So the endocarp is that inner lignified layer of your carpel, of your flesh. This is a lignified layer associated with your ovary development. This is the vascular bundle which feeds your ovary, which feeds your ovules, which feeds your developing seed. Because we're dealing with a droop peach fruit is a very basic fruit. It needs that seed to develop because it needs that developing seed to produce the growth regulators, which encourage the seed house, the carpel, the fruit to develop. Seed also needs a viable connection for nutrients, and lignification seems to be associated with that viability. So if it's a sort of a consequence that if we want fruit development, we're always going to have a little bit of a lignification. And unfortunately, this particular lignification is in the form of a lignified thorn, which is highly undesirable. Another interesting implication of this is that if you remember, each ovary has two potential seed. And typically with almonds, as well as with most peach varieties, one of those ovules aborts, and so we just have one seed development. Although it's out of sight, that abortive ovule also has its own funiculus, as shown in this particular slide. And with almond, we find when we see split pits with almonds, it's not at the suture seal, but it's almost always at this point where you have the degenerating funiculus. With peach, whether it's, and actually with prunus in general, whether it's peach or apricot or plum, we'll often see this asymmetry between the two sides of the suture. If you crack open the fruit and if you look at it, this undersized section is almost always associated with your abortive funiculus feeding that abortive seed. So in the last few slides, I'll show an example of some other traits we've been looking at. The other trait that we needed was brown rot resistance caused by Monolinia, particularly Monolinia verticula. This slide shows sort of a breeding history at the lineage of one of our breeding lines where the color indicates the amount of the susceptibility from lab tests. On the right side is cut fruit, so wounded fruit. So we're obviating the epidermis as a barrier. On the left side, unwounded fruit. I also want to point out that this is our firm free, the F8142 that we talked about earlier. This was a source for the resistance in this particular test. The other source was Bologna. But to emphasize that we've got good quality peach fruit, only 4 generations that after that initial hybridization between two very different crops, almond and peaches. As Xenia talked about, Bologna developed in the Pelotas area of Brazil, which has a very high rainfall, has very good tolerance to brown rot, but it's from multiple components and each of these components having small effects. So it's very difficult to breed it back. And a lot of those components, or a major component is a sort of a greenish epidermis, which once you breed out, obviates the epidermal barrier. Other components are things like a high phenolic content, which gives good resistance, but also increases its susceptibility to bruising. On the other hand, the almond source has given us very good quality fruit, and as you see from the individuals, fairly highly heritable. So much easier to transport, much easier to develop molecular markers for, as shown in this next slide. So this is a study by Pedro Martinez Garcia. And one of the advantages of having a genetic diversity is that you can, you have within peach, it's highly inbred. We have the domestication window. You don't have a lot of genetic variability. We don't have a lot of phenotypic variability. And both of those are prerequisites to get good molecular markers. By bringing in both a much more diverse genome as well as a much larger possibility of traits, it increases our ability to develop good markers as shown in this, where we've been able to identify the probable location of the major resistance gene and even the probable identity of it. The downside of this approach, by using novel sources of your germplasm, because that relationship may only exist in that particular lineage, that particular source, you need to develop molecular markers for each of your different sources. So it may be even if I use cultivated almond from Europe, it may be a different source than if I use cultivated almond from Persia, for instance. So it's more efficient, but more tedious in other ways. It's an interesting slide which shows some of the work with Jonathan Fresno Ramirez working with Tom Famula over in food science, where we use Bayesian analysis to try to characterize the inheritance of a major gene for fruit size. And this is shown in the slide. The gene that we're looking at, the Q, small q, and the small q doesn't indicate a dominance relationship, but it's just the way we designate it. So we've used Bayesian inheritance to try to predict how this is being inherited through the lineages, and then use the color code in terms of the fruit diameter, with the deep purple being the larger fruit. Again, going back to nonpareil as a source, as well as some of our traditional germplasm. Again, the almond source has proved a very useful and successful source for increasing the fruit size. And we believe this is by increasing the number of cell divisions early on in the mesocarp development, rather than swelling, which would preclude it to more susceptibility to softening. Interesting, that same analysis shows that some of the early founding clones for peaches, early Crawford, for instance, had this particular gene, but it was lost in its either the early breeding efforts, particularly in North America. The final aspect I want to emphasize is that oftentimes, very often, you can't really anticipate what the trait is until you bring it into a peach background, which are the desirable trait that ends up being desirable within a breeding program. And to show as an example, this is that Prunus Scoparia that I showed you at 9,000 feet on the mountain pass to Isfahan. You can see the very small leaves, early fruit development. Within a few weeks, these leaves will fall off. The stems tend to be fairly tender and green. The photosynthesis will occur here. In its native region, very little exposure to moisture. When we bring it to Davis for breeding, it's so susceptible to any sort of foliar and blossom diseases that we often have to cross it into a cultivated almond just to maintain it. You see very rapidly in the crosses back towards peach that it becomes peach-like within a few generations. And then once we get it into a background of peach, we notice that it was segregating for a major gene for, in this case, mildew resistance. Something we wouldn't anticipate from the origin, and something we couldn't really identify until we brought it into that peach background. Final slide, and Nikos had talked about one of the concerns. We have things like climate change, we have regulatory changes, we have consumer preference changes, but the scariest thing is this, what do we call these long jumps. You wake up one morning, you have an entirely new problem, and it's a very severe problem. An example is Sharka plum pox. This shows the processing peach production area in Ontario, which was a very important production region in Canada, decimated when Sharka came in, and it wasn't effectively controlled. Some of the only source that I know of Sharka resistance within peach were some of these peach almond breeding lines, which we had developed, were tested by Pedro Martinez Gomez in Spain, to the D isolate, and more recently by folks in France, where they found that the resistance was there. In France, they found it tended to break down into the field where they had other isolates as well. So a summary slide, related species offer a range of new traits, and by species, I'm including not only almond, wild almond, but also the wild peach. This slide shows some a dominant trait for pillar peach, which we brought in from some of the Prunus myra, which is a relative of peach accessions. The basic developmental constraints need to be recognized as we develop these new traits, these new options. A major limitation, because of the novelty of it, may be envisioning what opportunities are best amenable to solve the problems that we anticipate or the problems that we have. And this is particularly a problem because useful traits may not be apparent until they're integrated into a peach background. And finally, and again, I'm glad to be following Nikos's talk, because one of the major challenges facing all of us as breeders is developing options for the next generation. But with climate change, regulatory change, and the inevitability of new pests and diseases, we often don't know what those requirements are. By bringing in a lot of new germplasm into your breeding program, it gives you the options perhaps to more effectively address those. Thanks.
Thank you, Thomas. Great presentation. So many information, useful information for the breeders and for the ones that are especially interested in clingstone peaches. And Prunus species give us the possibility, Prunus genus give us the possibility to play with the genes, and it's wonderful when you can use some wild species to improve your activity, breeding activity. So the time is quite limited, but we have some questions here. If I may ask one from Joss Anderson. Thomas, has the stay ripe gene been introduced into fresh market peaches? Would it be advantageous for it?
Yes, and one of the questions I tried to address, so the fresh market peaches, the answer is yes, if you pursue the development of fresh market peaches using a clingstone peach, as is basically being done with all of the very early peaches, since they're essentially clingstones as well. As soon as you use a freestone genetic background, as soon as you use a freestone type peach, at tree ripeness, when that flesh, when the adherence between the flesh and the stone is removed, your connection between that flesh and its continued nutrition from the peduncle is also removed. So it's genetically programmed, I believe, to maintain its integrity, but it basically starved out by the disconnection with its source of nutrients. So inherently, the freestone doesn't give you the same options as a clingstone.
Do you plan to map the Statewide Trait? It's another question from Iban Eduardo.
Yeah, we're in the process of mapping it, and I should point out that we brought it in from several different sources, and we're looking to combine those sources. Almond has been one source. Another important source has been the South African germplasm, particularly the voltamate that derived from Walgant, which we believe are, which we've shown by that earlier study to be very unique germplasm. And also some of the Prunus myra has also been a promising source for it. So we have different sources, and we're trying to map all of those.
Okay, thank you. Please keep your questions for the last part of our session today. So, George,
Tom, there are several questions stayed unanswered, so you can make a sum up of them, and during the concluding remarks, you can raise your viewpoint for what you consider more important. There are many questions, both for you and also for Nikos. Okay. So we are moving to our last speaker of this session, Guglielmo Costa. Professor Costa served as faculty of agriculture at the agriculture of the University of Bologna. Professor Costa began his career with investigations on agronomic, genetic, and chemical methods to control vegetative growth in the main temperate fruit species. In this it is the use actually of plant growth regulators in fruit crop management that is probably the area of research for which he is best known in Italy as well as overseas. Other two Professor Costa major interests are the kiwi fruit research carried out since the introduction of the species in Italy, and the non-destructive assessment and prediction of fruit quality, particularly by the use of near-infrared spectroscopy and electronic nose. The results of the research in this field was the development of devices patented by the University of Bologna and now widely used in the industry. Among the numerous awards he has received, the most prominent is the ICHS Fellow Award in recognition of his outstanding contribution to horticultural science. The talk today is dealing to what extent chemical thinning can be applied in peach towards reduction of the cost. Professor Costa, the floor is yours.
Thank you, George. Let me, can you see my screen?
Yes, just press the presentation. Okay.
Yes.
Yes, you can start. Thank you.
Okay. Good evening, good afternoon, good morning, wherever you are. I'm going to share the results myself and Alessandro Botton from Padova University are obtaining on the peach chemical fruit thinning, where we are, where we can go. So let me start with this question: why fruit thinning is mandatory in peaches? We know that when you have okay, I would like to get the pointer, but anyway, I forget it. But anyway, if you look at the x-axis, the lower is the number of fruits, the less is the production, and it has increased up to totally per tree to a given point, and after it doesn't increase anymore. And the same thing is doing the fruit size. The less of the fruit, the higher is the fruit size, and is rapidly decreasing. Why the number of fruit is increasing? So in order to have a better fruit size, we can exploit the area of interfruit competition, and there is a fruit physiological drop that the tree is normally using to equilibrate the fruits to the production. But if we want to reach the higher optimum commercial yield and the optimum fruit size, we have to do thinning. So thinning is not just the size, but also the achievement of high fruit quality, and also maintaining a vegetative reproductive balance to avoid alternate bearing. This is mainly important, is more important in palm fruit, especially in apple. And when we talk about thinning, I think it will be interesting to do why in peaches the fruit chemical thinning is still problematic or complicated, while instead in apple fruit we had a lot of positive results. Why in apple we have a lot of chemical thinners that are available for, can be applied at the bloom thinner and fruitlet thinning. Since starting from the last century and the middle of the last century, ethylene, caustic chemical, ATS is a fertilizer, is not exactly a chemical thinning, but is used as a bloom thinner in several species. So fruitlet thinning, auxin, that are probably one of the most still reliable compound, and BA. And recently we have new molecule, metomytron ABA and SCC acid. I will spend some word about this new compound. But if you look at the apples, the decision support system practical tools that are available allow to make some interesting consideration. In the corimb, we have a king fruits that have a very low abscission intensity as compared instead to the lateral fruits that instead have a very high intensity as abscission. So that we want is to maintain the central fruitlets, the king fruitlets, and to avoid the lateral fruits. Also, we have a quite interesting relationship among the natural abscission wave. If the first drop is high, we know that the June drop will be low. On the contrary, if the first drop flower and fruitlets is not as expected or practically absent, we know that the June drop will be higher. June drop, of course, will be too late for having interesting fruit size. So there is a lot of algorithm, a prediction tool for apples that allow to tune up the chemical thinner efficacy. For instance, Duan Green come up with the green model that is measuring the fruit of the corimb and the fruit growing at the rate below 50% of the fastest fruit growing will upsize. Again, Malusim has been developed by Laxo, Terence Robinson, and Ali in USA, and according to the climate and the day average carbon balance, if it's high, you must increase the thinner rate. If it's low or very low, you must decrease or do not thin the fruits, because many fruit will fall off naturally. We in Ufring group are in developing some new device like these BPS1, so-called, for measure the leaf chlorophyll fluorescence, that allow us to understand what the stress induced by, in this case, the brevis thinning agent is, and for how long is this stress will last. And we come up also with algorithm is called BreviSmart, that practically, considering the diameter and the climatic condition before and after the application, gives some alert that is are in the yellow side. You must increase the concentration on, wait for a better phenological stage. If it's green, it's like a light. If it's green, everything is like you exactly define. If it's red, you must pay attention because you have a risk of overthinning. With Alessandro Baton, we are also come up with another prediction we call brevis spread, that practically take care of the hierarchy index. If all the fruit are growing at the same rate, the thinning that you can expect is lower. When you have a high hierarchy index, that means you have a differentiation between the king fruit that growing at the faster rate and the laterals that growing at the slower rate. You should expect to have a higher thinning predicted. We are also always with Alessandro carried some research recently to better understand the abscission physiology and to tune up the thinner efficacy. In this case, with the using of the thermographic camera, you must see that when you do application of the thinning agent, your fruits just one day after the treatment increase the temperature, and this may lead to a higher abscission. We are also in this last year just some graft, not a complete elaboration, using a hyperspectral camera to do some measurement. We go from 300 to 2500 nanometer, and the lateral, the king and the lateral fruitlet. You see, Alessandro, here that the taking the measurement is have a different spectra. We are also using, I just get this new indication, even Alessandro didn't see, it's the first time that it looked like it. So we will have to discuss in order to understand what the potential is of this. Eventually, if it can be used also on peaches, we use the kiwi meter. That is a this new device that allow the measure of the parameter, the index of absorbance difference that has been normally used to establish the ripening of the fruits at harvest. But in this case, we use on a very small fruit, and from 0 to 1.5, in this case on a golden delicious, this is the king, and this is the lateral. As you see, there are big difference between the spectra that the king is giving back as compared to the lateral. If we go to Fuji, we have to go in another class from 1 to 1.3 in order to have the difference between the king and the lateral. What doesn't mean this? We would like to be able to have the possibility to do another prevision of what could be the future abscission. So if we go to peaches, why hand thinning is the most common practice? Few chemical thinner are available. This is a very old slides. In apple, naphthalene acetic acid saving or carbaryl is not allowed anymore. In Europe, naphthalene acetic acid BA, and practically the only one that we had until now was ether, whatever you want to call, but several several other compounds has been tested. I tested a lot myself as well, 3-CPA, CZA, ordonyl, Moth, actins, etc., but practically only the ethiphon arrived to be used in the in some situation in the practice, or sometime on the clean peaches, but with a lot of problem, by the way. Why the peach is affected by cultivar? Cultivar play a very big role. Fruit developmental stage, natural potential of season, climate condition, chemical instability, for instance, from the only chemical thinner that was. So we have some practical rules. We did some flower thinning and fruital thinning. Flower thinning will be very interesting for the early ripening cultivar, since we load the competition just at the beginning, so we can increase the fruit size. That is one of the problem of the early ripening cultivar, or just for experimental purpose because it's too expensive. Fruit thinning is carried out when fruit reach 20 millimeter or seed length. Someone measured the seed length 9 to 11 millimeter, but the more important, probably some practical rules, like to leave a leaf to fruit ratio of 40-45 leaves per fruit, or to leave a higher number of fruit in the basal part of the shoots, or if the position of the shoot in the trees is exposed to the light, so you may have a higher number of leaves feeding the fruit, etc. Or genetic background is also has to be taken into account, because freestone peaches and nectarine require the stronger thinning than the clingstone. The algorithm, few are available. At the beginning, the time after full bloom, and thereafter fruit growth kinetics proved to be a more reliable phenological and practical parameter to establish the optimal time of chemical thinner. But again, if we look at the early ripening cultivar to compare with the very late ripening cultivar, as you can see, the pit hardening stage in the early ripening cultivar is difficult to be defined, and there are big differences, and so even the application time could be a problem. So there are some other models that have been studied, like consider the dry weight development, especially during the pit hardening stage, that was correlated with the final fruit size and quality. But some research pointed out that only under optimum carbon supply, so in late ripening varieties, there is an insufficient assimilate supply. This parameter is not reliable. So other growth models were established, representing the fruit growth divided into the 4 phases, and using first and second derivative of the double sigmoid curve. But as Angelo Ramina say, I spent several years working on peach thinning with Angelo. The variability observed among different cultivars represent a further complication and ample practical application of this model. Angelo came up with an idea that is practically a green model applied to the peaches. If the fruits that are growing at a very fast rate have a low abscission potential, while on the contrary, the fruit that are growing at a lower rate have a high susceptibility to abscise. So as a result, what we saw, in Ignasi yesterday with a very interesting presentation show us what is doing now more than chemical thinning is mechanical thinning. Mechanical thinning, of course, the grower has to buy the machine. The machine is not exactly cheap. You have to adapt the training system to the machine, and the machine to the training system. You can even apply on whatever method of production you are using. Organic standard is independent from the genotype, environmental condition. It can be portable, can be mechanical, carried out by the tractor, and someone like it, someone do not like it, but it gives some interesting results. Now there are new molecules that have been on the market in the last year and that we are testing. The metamitron brevis mimics the shading and inhibiting the electron transport of the PS2 system and reducing the photosynthesis efficacy up to 60%. And abscisic acid, the known well plant growth regulator, and more recently amino cyclopropane carboxylic acid, ACC, that is a precursor of ethylene, and I will spend some other word. We did some, I did some years ago, some testing with comparing the apples with the peaches. This is a application of the metamitron brevis. This is a control, and this is a reduction in net photosynthesis as you have here, and it lasts for several days. And this is enough for come up with a good thinning in apples. If you look at the peaches, you see this is a control and this is a reduction when you do the photosynthesis, when you do apply but the results were not so exciting like in apple. So the reasons are still going on, but so far, not so much. So there are two other compounds, as I say, ABA, abscisic acid, and ACC. ACC recently, the Valent Bioscience, and I thank Steve McCartney for having sent these slides, is expecting the registration by US EPA in this year. The compound will be called Laxid, as you see. It's the ACC synthesis and metabolism in plant is just the immediate precursor of ethylene. Is not aggressive like ketophone, so no gummosis, and is not so affected, is not so strongly temperature dependent. And we also tested in the UFRIN, European Union Fruit Research Institute network, is it's a group of mainly European researchers, ACC, at Peter Fall. We get some interesting results as a thinning at the beginning and with a higher concentration even at harvest. Why ABA, the effect more or less follows the same thing. The higher concentration was affected, but the results was not as good like ACC. ACC did increase, give a possibility to increase some fruit size. ABA not exactly give just a better distribution of the compound. There is also, since the UFRIN is an European, but we have also some people from out of Europe, like Karen Theron, for instance, from South Africa, that every year is join us in our meeting and it did some trial with ACC, comparing different concentration of different cultivar. Let me show this, and he pointed out that there is an effect. The more you increase the concentration, the more there is an effect. We have to pay attention, because if you're higher concentration, you can cause some over-thinning. Of course, you can increase also fruit size, but you may increase, you can have a risk of over-thinning. What the indication from the Valent are that the fruit number per shoot and the hand thinning are reduced, can reduce the number of fruit by 14%, and this lead to more or less 30% of the saving when you have to do the hand thinning. They say you acid plus hand thinning. And these are the recommendation from the company for peach and nectarine and plum, and also for apples. Let me point it out that for apples, these as you can see, the timing can go from 6 to 25 millimeter king fruit diameter. And that, if you know, there are very few thinners that are affected at so large fruit diameter. Some years ago, or still now, we were looking at a different approach. So the idea was to call strategy. Strategy is a definition that is normally used for apples, since they have several, apple can count on several thinning agents. They may apply with the bloom thinner like Etefon, and after they can use Oxin, and after they can use BA, and eventually after they can use Brevis, etc. And in peaches, we do not have all these options. So we thought that instead of using several thinning agents, we could, for thinning, we could consider something to affect the fruit load, to control the fruit load. For instance, applying gibberelling, we did some years ago, as you see, been published in 1996, myself in 2000 with the GA, and during can inhibit the flower bud formation. There was at that time a compound under the commercial name of release, and now there are some new development by other companies as well that are interested in this possibility. Or you can have a possibility to see what the results are. You can arrive with some other compound like dormancy breaking agents that can be applied to interrupt dormancy, even cause some bud abscission. In this case, look at the GA applied in May or mid-June. This is a control 32.64, or you can reduce 21.17 the number of flower per shoots. And with the dormancy breaking agents, you can go from the thinned control or 19 or 15, and you can cause a 41% of bud abscission if you decided that you need another reduction. You have these a trial carried out spring test. You see the untreated control, the treated, and the difference in fruit size that it was possible to obtain. These some other compound that's been tested by Rygard, Greg, and Ismail Falahi in South Carolina and Idaho. Percent of bud kill or percent of unopened. So this is interested. Sometimes you do not get exactly the results you are expecting. In this case, you have more flower to the top of the shoot as compared to the distribution that eventually is just the opposite of what you will like, but you have also the possibility to cause some thinning. Thinning a bloom, ATS or endothel or well thin or thinix several, and thin it 19 when you do apply is 30%. So is already the third treatment. Of course, your flower do not look very nicely, as you can see. This control and this is treated, look more like winter or a cold damage, but it is not. And finally, you have a last option that is a fruitlet thinning. So Etefon, I don't think that a lot of people still using Etefon. ABA, probably not, but acid, as I showed before, could be another option that can arrive to tune up all the chemical thinning in peaches. Is finished? No, it's not finished, because we have to consider the climate change. Temperature is increasing. The minimum temperature are expected to rise more rapidly, causing a reduction of winter chilling in warm growing area of temperate fruits. So the winter chilling accumulation, as we know, is essential for exiting dormancy, and is a major factor that determines the suitability of areas for many commercially important fruit crops around the world. We are facing now a climatic condition, will be probably climate change, a climatic condition that are exceptional. This year, for instance, in Italy, we had the winter chilling accumulation, but the accumulation just occurred in the last 2-3 weeks, not during the winter, but almost at the beginning of the spring or just immediately before the spring. And this also can make a difference. So a lack of insufficient winter chilling has to a negative consequence on fruit thinning, because you may have different phenological stage in the same traits. An advance or spring event or a delay of the autumn event, again, as to causing a modification of the most orchard management practice, and thinning is one of these. Rainfall, sometimes it has never rained. When it decided to rain, it never quit. If it does during pollination, you have a reduction of fruit load, and the thinning operation has to consider also this. So it's difficult to decide when to do the thinning or not. So what are the future perspective of thinning in peaches? We know that fruit abscission is a complicated physiological processes that influenced by a series of factors, internal and external, that complicate the scenario. The climate change further increases this complexity, but we have to consider that the grower expectation toward the future technological advance in fruit thinning are enormous, since the income that the fruit grower has from the peach orchard are totally unsatisfactory. Production costs do not cover the fruit thinning, represent one of the highest costs. So these, as a result, the best area where we were growing peach are been reducing. Look at the reduction of the peach in Europe. This could be a problem for a very marvelous fruit like peaches. So what the future perspective? Establishing an international platform for communicating results of horticultural research within the whole productive chain could represent one of the most immediate solutions. We have the UFRIN working group. It's called UFRIN Working Group on Chemical Thinning, mainly deal with thinning, but there are Northeast Plant Growth Regulators in USA, or the ISHS Plant Bioregulators Working Group. And these are the actors that we are producing every time that we are doing one of our meetings. So there are this group, but this group are formed on the voluntary basis and do not receive any institutional fund. From one side, it's true that the existing network can guarantee an active and dynamic open discussion on thinning topics, but such a networking should be supported by a structural funding policy with a long-term view. I hope that is not to be too pessimistic, and I hope you are an optimistic person. And as you see, you may agree with the three sentences that may scare us, but when we saw who say something like this, eventually it's probably, was 200 after Christ. So this was already one of the. Thanks for your attention.
Thank you, Mimo. A great presentation with so many question marks on so many factors that are affecting the peach thinning and the peach physiology. So it's not easy, as you mentioned, because of so many problems related to the final fruit cost at the farm gate, and so many other problems that the producers are facing nowadays.
Florence, we have a question from Juan Carlos Melgar. Have you seen cultivar differences when using Axid? He used at 300 parts per million in 4 cultivars this year. I did either 1 or 2 applications at 20-25% bloom, and only saw some effect only in 1 cultivar. So it is cultivar dependent, this application?
Peaches, the cultivar are affecting extremely the thinning results. This is why we are thinning apples or even pears, but very little of peaches. There is the cultivar play a very important role in susceptibility to be thinned or not. Probably the fact that everybody is a single individual, you know, so everyone is competing with the other bud, and in every node you have a flower bud and two a shoot and two eventually flower bud is another matter, but is complicated. The competition in peaches is strong, but even in apples, but the main competition is between the king and the lateral flower. And when the competition is on, it's easy to thin. When all the fruits, as I say, are growing at the same rate, it's not so easy. Another aspect is that the peach is very light dependent. So peach is probably a species extremely efficient, and it's not so easy to come up with a practical, with a thinning agent that can be easily transferred to the practice. This is a story. As far as I know, I started to work on chemical thinning of peaches very long time ago, and I didn't solve yet. So not just me, but all the people working on peaches, you know, or we are causing a damage like a caustic agent. And in this case, it's possible to, even if the trees does not look very happy when you apply this compound, you can come up with some interesting results. But why have not been transferred to the practice? The people is afraid of the frost damage. If you reduce the number of flower, or if you damage the flower, the people say, what happens if a cold damage arrive? If the cold damage arrive, it certainly cause damages, but it depends how many degrees below zero, it depends for how long, it depends your condition, and if nothing happened, you have to do hand thinning, and the hand thinning is very costly. So I can believe that there are differences between cultivar, and the more the cultivar is early, the more it's difficult to be thin, because you do not give to the tree the possibility to adjust itself what the vegetation reproduction relationship.
Lauren, any other question by the attendees?
It was just a comment on
cultivar difference. They confirm a colleague from United States that yes, there is a difference among cultivars.
On oxide.
I would like to ask Christos to spotlight all speakers of the day. It's a difficult Friday night in Europe, so still we have many attendees still being active, so this is much appreciated. I have seen that Tom Gradziel actually respond to all questions, but I was wondering if you would like to highlight anything based on the questions that you have responded that would be important for all attendees.
Sure. I guess if there was one message, and again, it says the previous speaker, responses will differ by cultivar and also by fruit type. And so we have the freestones, the clingstones, the melting clingstones, the melting freestones. An interesting possibility, again, to apply some of these things we've been developing with clingstones is the very early freestone market, where many of the breeders have in fact been utilizing clingstones because in this early maturity, you don't really get the separation of the freestone in any event. Another possibility, which is intriguing, but I haven't, which could be followed up on, is I remember in the past, Bruce Topp and Wayne Sherman's program had identified a type of peach which was intermediate. And so it was a semi-free type peach, where it was essentially a freestone, but as that stone separated, as you physically separated it, you would see the adhesion of some of those basal vascular bundles. And so you have the possibility perhaps of getting the best of both worlds, of maintaining that connection, feeding your fruit as it gets into that multiple weeks after over ripening, after full tree ripe stage, but also getting that freestone and maybe some aspect of volatilization to give you that characteristic freestone flavor. So I'll leave it at that.
Excellent, excellent, Tom. Laura, any concluding remark from the questions that you have responded on private mode?
Yeah, there were a couple of questions, but I think they were very technical about the range of chilling requirements in the PeachRefPop and the locations as well. So I mentioned in the location, we go from 700 to 1,700 chilling hours. The chilling requirements are ranging between 50 and 1,200. So we have a large range, and we think this is very interesting, also in light of changing climates, changing temperatures, and having these really different locations will be very interesting to do the study on phenology. So I think this is one of the examples of how such an experimental design can be used to really understand the environment, genotype by environment interaction.
By the way, I have promoted Professor Bassi as panelist. So Professor Bassi, if you would like to add, since this was a cooperation with Laura, if you would like to add something more on what you did as a group.
Thank you, George. Not really. I guess Laura was really exhausted in her presentation, so I have no more, unless there are additional questions from the audience.
Okay, okay.
Have you used, Laura, a standardized phenotyping? It was one of the questions.
So this is really a good question. Actually, that was one of the main questions I saw in the chat. For sure, standardizing phenotyping methods is always a challenge, as the audience commented. In previous projects like food breedomics, there was a big effort to agree on these standards. So, for example, in that case, I remember we had many discussions, and in the end, it's always a matter also of experience. The people that have a lot of experience in a trait can say, guys, do it this way because this is the perfect compromise between the throughput and the robustness of the data. In the case of the PeachRefPop work we are doing, let's say that it was something, let's say, new. So our group proposed a phenotyping protocol, and this was adopted by the others. And vice versa, there's always some exchange. When people go and try the protocol in the field, they feedback with some challenges, and so the group has to really strongly coordinate. This is the key.
Francois, you cannot just open the microphone.
If I may just give our experience on Invite, because we're doing the same on the Apple. I think we're probably a little bit ahead. Hello, Daniele. So we started by, so we have already 3 years of assessment of fruits, but it's very basic assessment. So we just weigh the fruit, we look at the color. Actually, we look just at the pack out things. Some sites are looking at the sugar and things like that. But what we're starting, we started last year and we'll develop in the next few years is to use a high throughput phenotyping system with drones and things like that. So we'll try to implement that, because actually in all genetic or even breeding, at least on crops, the bottleneck is the time we spend on phenotyping. So we really invite the full work package dedicated for that. So if you, Laura, I propose you or Daniele to exchange. I think it's a way, for example, to exchange with you. The idea for us is to use a very cheap device to take photo and to analyze the photo. So this is what we are doing.
Thank you, Francois. I would like, Laura, would like to add anything or? To
no, that was one of the questions we had.
Excellent.
Also, we discussed in our group to use drones and to use other types of approaches. So this is one of the things that we are interested in hearing also other people's experience for sure.
Very, very well.
The problem is not the draw, nothing like that. The analysis of the data, the models you develop, the deep learning behind. It's
Sure.
Yeah. Florin, we're waiting for your concluding remarks.
Yes, so 4 speakers, actually 5 speakers, including Francois Laurens, with outstanding presentations, with so many details on hot aspects of peach research and also production. So we received so many questions, and having still this huge audience from all over the world means that our session today was extremely successful. And I want to thank all the presenters for their excellent job today. In my position of ISHS Peach Working Group Chair, I would like to congratulate George Manganaris and all the Greek team for the idea of organizing this webinar, that for 4 days keep the peach world researchers and not only together. In the same time, it was a wonderful introduction for the next year event in Greece. And what I would like to ask to all the attendants is to invite them, not to ask them, but to invite them to join us all in the next year Peace Symposium in Greece. I'm sure that our Greek hosts will do a perfect work to organize these important scientific events in peach. So Nausa, it's a perfect place, and the organizing team, it's also a very, how to say, efficient team, and I'm sure that it will be a wonderful event next year. We should not forget, as an ambassador of International Horticulture Congress in France, after all the experience in Greece, in the wonderful country of and the area of Nausa, we should all move to France, because in Angers we will have the way to, how to say, to compare the feta cheese taste and the ouzo and the rakia with the French cheese and wine, and to meet our wonderful French host there. So I think 2022 will be a very intense summer. So all of you are invited to join us both in Greece and in France, in Angers. Once again, from my side, all the congratulations for this important work, and I hope to meet you in person, both in Nausa and in Angers. Thank you so much, George.
Thank you, Florin. I would like to extend my gratitude to all speakers, and of course to all attendees. We received 760 registrations, and on average, we had from 200 to 300 participants, attendees per talk. So we have a good pool of people who have proven interest in peach. And of course, we're looking forward to welcome many of them in Greece next year. I will update all attendees with all our research activities in the future, so as even if they are not able to attend the meeting in person, to have an update about recent advances about peach fruit production. Thank you very much, everybody, and have a nice weekend ahead.
Thank you, George.
Thank you very much.
Bye.
Thank you very much.
Thank you.
Congratulations.
Thank you very much.
Congratulations. Very nice.
Grazie.
Thank you very much.
Ciao, grazie.
E fa di sto.
Thank you. Bye bye.
Thank you.